Guess I'll get started. So good afternoon, everyone. My name's Gerald Bush. I'm a member of Dr. Dan Liebel's lab, and today I'll be giving my presentation on reactive glial D-Serine regulates synaptic recovery following traumatic brain injury. So every year it's estimated globally that approximately 75 million people will sustain a traumatic brain injury. The CDC reported in 2020 that there was approximately 200,000 TBI-related hospitalizations, and in the same year it was reported that there was 65,000 TBI-related deaths. So if you look to this graph on the right, majority of TBIs that are classified in the clinic fall into this mild category, about 82%. So this would be things such as concussions and lighter head injuries, sports-related accidents, and things of that nature. But about 10% of TBIs fall in this moderate category. So this is, and a lot of these are attributed to motor vehicle accidents as the major causative for that. So my talk will be really focused on this moderate category. So traumatic brain injury can be classified into two distinct phases of injury. First, the primary injury, and this is a result of the initial mechanical impact to the brain. So it's really a focal, all the damage really occurs at that focal site, at the site of injury. And kind of the injuries we see here is tissue death from the mechanical stimuli, hemorrhaging, axonal injury, compromised blood-brain barrier, and blood vessels. But over time, from seconds to months to even years after this primary injury, we have the secondary injury. This is a diffuse injury where it can affect structures distant from the penumbra. And really these injuries are a result of the cellular response to that initial physical trauma. And some of these insults include things such as gliosis, mitochondria damage, oxidative stress, cell death, alterations in glucose metabolism. And really what I'll be focusing on is the synaptic damage that is a result from these secondary injuries. So in a longitudinal study that followed TBI patients, what this lab did is they assessed at 2, 5, and 10 years post-injury different domains of cognition. So they looked at areas in executive functioning, speech, and most importantly, learning and memory. And what they found was that about 65% of patients in the moderate and severe category reported issues 10 years after their initial trauma. So this would include things as memory problems, difficulty in planning, concentration, and slower speed of thinking. So this just really highlights the importance to really start looking into research options to mitigate these long-term effects. So what are the current treatment modalities that are implemented to kind of for traumatic brain injury? So really the current treatments really just focus right now on stabilizing the patient through surgical means, relieving intracranial pressure, or pharmacology. There are actually no current therapies to really look at these long-term cognitive deficits that are traumatic brain injury. So once again, just highlighting the importance of we need to develop strategies to address these issues that greatly impact quality of life. But before I go further, I kind of want to give what my talk will be about. So really what my focus is on, I'm going to be giving a lot of, intro slides of how I got to my question. But my overall question is, how does D-Serine modulate synaptic recovery following traumatic brain injury? And these next few set of experiments were done by a previous graduate student in our lab, Emmanuel Perez. So how do we model a moderate traumatic brain injury in our lab? So we use the controlled cortical impact model to induce brain injury. And this is performed where we anesthetize the mouse, we perform a craniotomy, expose the cortex, the dura is left intact, and then a piston delivers a fixed blow to the brain at 4 meters per second. But really what I want to highlight here is within our model, we see at 7 days post-injury, there's extensive cortical damage. However, the underlying structures such as the hippocampus remain structurally intact. But when we went to kind of quantify what's going on in the hippocampus, we want to look at, is there any neuronal death? So when we looked at that, we looked at these different neuronal populations in the hippocampus, the CA1, CA3, and the dentate granule cells, and we actually saw no significant neuronal death. However, when we assessed kind of the synapses within the CA1, through electronic EM images, what we found actually is there's a significant loss of synapses within the CA1. So what does this all really tell us? This tells us that we have an excellent model to study synaptic loss in the absence of neuronal death. So next, we also wanted to start kind of characterizing our model like functional changes. So first we performed electrophysiology, where we recorded from the CA1 of the hippocampus and stimulated the Schaefer collaterals. And what you can see, this was done at 7 days post-injury, and what you can see in the white open boxes are sham animals. And following a high-frequency stimulation at 30 minutes, we're able to induce long-term potentiation, or LTP. However, in our injured animals, these black solid boxes, we see we are unable to induce LTP. This would suggest that this is due to this loss of synapses within our model. So before I go further, I want to give us more background on the NMDA receptor, as that's going to be important for my talk. And really, the NMDA receptor is composed of 2 GluN1 units and 2 GluN2 units. These GluN2 units, or the GluN subunit, really comes in a variety of different flavors. But what I want to focus on is these GluN2A and 2B subunits, what I'll talk about later. So we need two conditions, really major conditions must be met for the activation of the NMDA receptor. First, we need binding of an agonist and a coagonist. The agonist would be a glutamate, and historically it was believed that glycine was the coagonist that would always bind to these NMDA receptors. But recently it was identified that in areas in the brain involved in learning and memory, such as the hippocampus or amygdala, D-Serine is actually the preferentially bound agonist, coagonist. So once this first condition is met, next we need a depolarization of the membrane to kind of release this magnesium block. And this is done through AMPA receptors. And once these two are done, then calcium influxes into the cell, which will result into increases into prosynaptic factors such as BDNF and K kinase 2. So next we need to understand the biosynthesis of D-serine. So in physiological conditions, the biosynthesis of D-serine really begins with glucose. Glucose is uptaken into astrocytes where it undergoes biochemical reactions and it's converted to L-serine. Once L-serine is transported out of the astrocyte and then uptaken into the neuron. And then a key enzyme, serine racemase, in the neuron converts L-serine to D-serine. And this enzyme is critical for my talk as I'm going to be using serine racemase really as a proxy for D-serine in upcoming experiments that I talk about. So next we wanted to provide further evidence that neuronal D-serine is the major contributor in normal synaptic transmission in physiological conditions. So to do that, we performed another electrophysiological experiment where we generated an astrocyte knockout of serine racemase in the blue boxes and a neuronal knockout of serine racemase in the green boxes. And what you can see with our controlled animals, this was all done in naive conditions, uninjured conditions. And what you can see is following the high frequency stimulation at 30 minutes in our wide open boxes or control, we're able to see the induction of LTP. And that's also mirrored within our astrocyte knockout model. However, when we look at our neuronal knockout model, we actually are unable to induce LTP. This suggests and provides further evidence that neuronal serine racemase is the main contributor of D-serine in physiological conditions. So next, kind of give a quick recap of everything that I've talked about. Is really and the schematic is showing a presynaptic, a postsynaptic cell where glutamate is being released presynaptically, as well as D-serine is being produced and released neuronally. It then binds to these synaptic NMDA receptors, which then leads to downstream signaling, which promotes synaptic learning, synaptic formation, and learning and memory. And these astrocytes play kind of a supportive role, kind of uptaking that excess D-serine. So what happens in our injured conditions? So to look at that, we did some immunohistochemistry experiments, and we wanted to look into the hippocampus of what's occurring. So in these top 3 boxes is our sham conditions. And you can see in purple, we have our serine racemose stain. And you can see that serine racemose is really localized to these neuronal populations in the hippocampus, such as the CA1, CO2, CA3, and the dentate granule cells. We also have stain for astrocytes and GFAP and microglial cells and IBL1. And you can see these are just kind of heterogeneously scattered throughout the hippocampus. However, following injury, we actually see a significant increase in serine racemose expression. Now, it's not just localized to these neuronal populations, but also spread out through different areas, such as the molecular layer. And we also see these increase in these reactive astrocytes and microglial cells. So next, we wanted to investigate kind of what is the consequence of this upregulated serine racemose in these reactive glial cells. So to do that, in our first experiment, we performed contextual fear conditioning. This is a hippocampal test for learning and memory and how it's performed. It's a 3-day assay where on the first day we perform a 10-minute habituation. The second day there's a cue and then a shock is delivered to the animal. And on the third day, the mice are placed back into the apparatus and then we assess freezing, where increased freezing is indicative of the animal being able to recall the area this aversive stimuli was delivered. So first we wanted to look at kind of what happens if we remove this astrocyte D-Serine. So if we take a look at our control animals, our serine racemose flox at 7-day post-injury, compared to our sham group, we see that reduction in freezing. However, when we take a look at the astrocyte knockout of D-Serine, we're able to rescue that deficit, meaning that removing this astrocytic D-Serine leads to the animal being able to recall this area where they were shocked. We also did this in microglial cells, which also have been shown to upregulate serine racemose. And we used 2 different knockout models. We used under the CX3CR1 promoter in microglia, as well as the TMEM119 promoter. And you can see in our control animals at 7-day post-injury, the deficit following in our contextual fear conditioning assay. However, knocking out microglial D-Serine and either of the promoters led to a rescue of this deficit. So next we want to take another approach, an anatomical approach, to look at what is happening to these spines. So to do that, we performed a Golgi analysis. This is a silver impregnation stain that stains the dendrite. And really the 2 morphological variants of spines that we really want to focus on are these mushroom spines, which are more indicative of kind of healthy functioning spines, and also these long, thin spines, which are indicative of these degenerative spines. And what we see is in our control animals, we see a reduction of these mushroom spines, a loss of these healthy functioning spines at 3 and 7 days post-injury. But in our microglial knockout and in our astrocyte knockout, we're able to rescue the loss of these mushroom spines at 3 and 7 days post-injury. And the thin spines, we see at 3 and 7 days post-injury, we see a significant increase in these spines, suggesting that there is damage occurring in the hippocampus due to the increase in these thin spines. However, once again, when we remove microglial or astrocytic D-Serine, we're actually able to prevent this. So this is all starting to kind of paint a picture that removing just one of the glial-derived D-Serine sources really can prevent this synaptic damage that we see. So to further this point, once again, we performed another electrophysiological experiment where we assessed the astrocyte knockout of serine racemose. And if you look at 30 minutes following a high-frequency stimulation, we see that in these black boxes are injured control animals. We see we're unable to induce LTP. However, in our astrocyte D-Serine knockout model, we're able to see that we're actually able to rescue this and induce LTP, against reinforcing that idea that removing one of these D-Serine sourced, glial D-Serine sources can prevent this synaptic damage and cognitive deficits. So in summary, and under injured conditions, what we see is an increase in expression of serine racemose in microglia and astrocytes. This then leads to a large release of D-serine, which acts on synaptic and perisynaptic NMDA receptors, which leads to hyperexcitation. And we've shown that we can preserve these synapses by removing either of these sources of glial D-serine in our knockout model. So the next step is we're going to look at what is the mechanism mediating glial D-serine induced synaptic damage in TBI. So majority of this work was done by another previous graduate student, Dena Arazanovska. And in this schematic, what we're looking at is the quadpartite synapse, where we have a presynaptic membrane, a postsynaptic membrane. We have the microglial cells in orange and the astrocytes in purple, where in these red dots, glutamate is being released presynaptically and acting on these NMDA receptors in the synaptic cleft. We also have these orange NMDA receptors, which are these perisynaptic NMDA receptors, and in physiological conditions, they don't seem to really play any significant role. Really, they're only active in pathological conditions, such as traumatic brain injury, Alzheimer's, or ischemia. So activation of these synaptic NMDA receptors is really like the mainstay of synaptic transmission, where we see activation of this leads to the phosphorylation of Krebs and then promotes other synaptic strengthening and growth factors. So what happens in a scenario where we injure the animal? What changes? So now we see these glial cells, microglia and astrocytes, now producing D-serine, and this D-serine then binds to these synaptic NMDA receptors, which contain this gluN2A subunit, the healthy subunit you could say, and then also binding to these perisynaptic NMDA receptors, which contain this pathological gluN2B subunit. So really we're seeing an enhancement of both of these type of NMDA receptors. And we're starting to see that activation of 2B seems to block the 2A signal. So I'm going to show you a scenario where if we knock out glial D-serine, we actually affect the signaling that occurs in the postsynaptic membrane. So if we look at our control animals, the serine racemose flox animals, we see that we evaluated a sham 1 day post-injury, 1 DPI, and 3 days post-injury. And what we see is that at 1 day post-injury, a significant increase in our gluN2A NMDA receptors as well as the gluN2B, suggesting that D-serine really upregulates overall NMDA receptors following TBI. But about 3 DPI, this kind of goes down. This goes down, and this could be due to just synaptic loss that's occurring in our models or other factors. But if we knock down glial D-serine and microglial D-serine, what we see is that 1 day post-injury, we're able to prevent this overall upregulation of these NMDA receptors. So we also wanted to look at the activated form of gluN2B receptors. So we did this by looking at phosphorylated gluN2B. And what we see is that in our control animals, at serine racemose flox, at 1 day post-injury, we see a significant increase of this phosphorylated gluN2B subunit, suggesting that these gluN2B NMDA receptors are increased, but also in an active form. However, in our microglial D-serine knockout, we actually see a significant decrease in our phosphorylated gluN2B subunits at 1 day post-injury. But at 3 days, it increases. So it's important at this 1 day post-injury because majority of the synaptic damage occurs at this time point. So this is really telling us that D-serine is functioning to activate these perisynaptic receptors, which leads to, and these NMDA receptors, which increases global expression and activation. So we also wanted to look at other factors, such as other factors in signaling. So we wanted to look at phospho-Creb and BDNF. So what we found is that in our control animals at 1 day post-injury and 3 days post-injury, we don't see a significant increase in phosphorylated Creb. However, we do see in our microglial knockout at 1 and 3 day post-injury a significant increase in phosphorylated Creb. So what is this really telling us? This is telling us that activation of these perisynaptic NMD receptors really plays an inhibiting role of preventing normal synaptic transmission by these gluN2A subunits at the synaptic NMD receptors. And this is also further shown in the BDNF, where we see in our control Serine flox animals at one and we don't see expression of BDNF until 3 days post-injury. However, in our microglial knockout of D-Serine, we see a significant increase in BDNF at 1 day post-injury. Once again, suggesting that by preventing the activation of these perisynaptic NMD receptors really ameliorates these deficits we see. We can now there's normal synaptic NMDA transmission propagating, which then leads to the activation of prosynaptic factors such as BDNF and other transcriptional factors. So next, we wanted to ask another question. So what is the role of these perisynaptic NMDA receptors? And another way to look at this is using pharmacology. So what we used is romaliate, which is a selective gluN2B NMDA receptor inhibitor. And what we see if we look at these vehicles at 3 days post-injury, we look at these mushroom spines, and once again, these are those healthy functioning spines. We see a significant loss of these mushroom spines. However, in our thin spines, we see at 3 days post-injury, a significant increase in these thin spines, suggesting that there's degeneration occurring. But when we use a dose of this romaliate from 1 to 100 nanomolars, we're actually able to augment the damage. We no longer have the damage like we see in the vehicle. So at 3 days post-injury, we see a rescue of those mushroom spines. And the thin spines, we prevent the increase of thin spines. This is just another way showing that inhibiting these perisynaptic NMDA receptors will ameliorate these deficits in learning and memory. So next, we wanted to do a loss of function study. So we used a knockout model of gluN2B. So this knockout model is a neuronal knockout of GRIN2B, which is the gene name for gluN2B. And what we see in our control groups at 7 day post-injury in our mushroom spines, we see that we lose a significant amount of these healthy mushroom spines at 7 days post-injury. But when we knock down gluN2B and neuronally knock down gluN2B, we're able to rescue these deficits we see. We also want to look at this contextual fear conditioning, and we saw similar findings where in our 7 day post-injury control animals, we see there is a significant decrease in freezing, suggesting that these animals no longer can remember this stimuli. However, at 7 days post-injury, in our neuronal knockout of gluN2B, we're actually able to prevent this deficit. Once again, adding further information that really these perisynaptic NMDA receptors are playing a huge role in this synaptic damage. So next, I want to go into the next step in the mechanism of damage. So what we wanted to focus on is phosphatidylserine. PS is a marker for cell death, and it's usually exposed into the inner leaflet of the plasma membrane. However, some stimuli can cause this PS to flip internally to the external membrane, which then, once it's externalized, it can act as a marker where its complement C1q binds to it, and then Once it's bound, we have the eat me signal. So in our model, our hypothesis is that is overactivation of these perisynaptic NMDA receptors from these glial levels, from glial levels of D-Serine really leads to this high increase of calcium influx into the cell. This calcium flux then causes PS to externalize, allowing PS to bind to C1q for phagocytosis. So to show that, and to visualize phosphatidylserine, we used a dye called PSView. And it's important that this dye can only bind to PSView phosphatidylserine that's been externalized. It cannot cross the plasma membrane. So we see that most of this PS staining is really done, and you can see it's localized to these areas of increased damage. But what we're really focused on is the area in the hippocampus, which is the molecular layer. And next I'm going to show you some kind of high magnification of areas in the hippocampus. So in this top image, in this study, what we wanted to really look at is phosphatidylserine really co-localized to these synapses. So to do that, we used a presynaptic marker vGlut1 and a postsynaptic marker PSD95 in green, and then PSView. So in the top group, serine Erasmus, and our control group, we see really just a co-localization of these pre and postsynaptic markers, and very little, if any, of the PSView stained of the phosphatidylserine, which makes sense since there's no damage occurring. However, at 3 days post-injury, we see a significant increase of phosphatidylserine. And we also could see at even a higher magnification that this phosphatidylserine is co-localized with presynaptic and postsynaptic markers. However, if we eliminate D-Serine, microglial D-Serine, we're actually able to prevent this flipping of phosphatidylserine from the inner to the outer leaflet of the plasma membrane, as seen here. So next we wanted to assess C1q, the binding partner for phosphatidylserine. And this is very similar images to the last one, as we see in our serine Erasmus flox control animals, sham animals. There's no really expression of the C1q. However, at 3 days post-injury, we see a significant increase of this C1q stain. And it's also co-localized with these pre and postsynaptic markers. However, at 3 days post-injury and our microglial knockout, we're able to prevent the increase of the C1q. Once again, suggesting that removing glial D-Serine plays a role in preventing synaptic damage. So pretty much in summary, we've seen that it takes both of the glial cell types, these reactive glial, microglia and reactive astrocytes, to lead to synaptic damage. Both of these cross a threshold that elicits synaptic damage. However, eliminating microglial or astrocytic D-Serine leads to synaptic preservation. And what's really next unknown is what is occurring in the synaptic recovery phase. And that's something my project is going to be looking at. So the questions I'm going to be asking is, how do neuronal or glial source D-Serine influence synaptic recovery? Is it beneficial to have D-Serine levels elevated above physiological levels? And 3, what happens when there is subthreshold level of activation? So to do this, I'm going to be using 2 different mouse knockout models, where I'm going to be assessing them at 28 days post-injury, a month post-injury, and I'm going to be inducing the knockdown at 7 days post-injury. And this is to really allow all that synaptic damage to occur. And then we can see what happens if we remove D-Serine. So to really look at neuronal D-Serine, what is its contribution to synaptic recovery? we generated a double knockout of serine racemose and astrocytes and microglial cells. So knocking this out allows us to just study how is neuronal D-serine modulating synaptic recovery. To assess the contribution of these glial cells in synaptic recovery, we're going to be using a neuronal knockout of serine racemose. And here we'll, here we'll be able to assess how does neuronal D-serine contribute to synaptic recovery. And then from here, we're going to be doing some downstream analysis, just looking at fear conditioning, biochemical analysis, Golgi analysis, kind of. So for our next question, is it beneficial to have D-serine levels elevated above physiological levels? We're going to do a gain of function study, where once again, we're going to, in wild type mice. So at 7 days post-injury, we're going to infuse these animals with an osmotic pump that delivers various doses of D-serine. And from there, we can perform our downstream analysis to say, is this beneficial to have elevated levels of D-serine following an injury? Also to look at this, what is going on, what is the influence of this sub-threshold level of activation? We're going to do some inhibitor studies using bromeliate as well as memantine, a more global NMDA receptor inhibitor. So next I'm just going to leave with like a hypothesis of what my studies are going to be. So I think elevated D-serine may play a beneficial role for modulating synaptic recovery. So this is kind of an intro of what I'm going to be doing in my project, as I had to change to a new project. So right now I'm at the mercy of the mice, waiting for them. So that's it. Thank you for your time. I'd like to thank my lab, my committee, the neuroscience program. Thank you. I think we did that in-house. Yep. Yeah, they're surviving. I got some under surgery finally. So they're just getting their first round of tamoxifen treatments now. No, I have not actually. So I'm generating those right now. So hopefully after some behavior in about 2 weeks, I'll be able to get those ready for Golgi staining to look at that. It's Yeah, well, we're gonna see all this damage, so mhm. Good question. Good question. I'll have to look into that again. We have someone who helps us out for genotyping, so I'll have to definitely look into that. Double check that. Yeah, yeah, because that makes sense. Yeah, both have it. Hmm. Yeah, I'll definitely get an answer for that. Yeah. Yeah, that's kind of interesting. So what's kind of, if we look back to like developmentally, in development there's actually D-serine is released, like is increased in is released by astrocytes to promote synaptic formation and little elevated levels above physiological levels during development. So my hypothesis is kind of based on that maybe just not just sub threshold levels of D-serine may actually promote synaptic recovery in some form. Yeah, yeah, yeah. Not over the threshold level. So there's a threshold level that occurs. So if we go over that, it leads to synaptic damage. But if we, my hypothesis is based on kind of development where there's a slight increase of D-serine, not at this detrimental level that leads to synaptic damage, but if we get to that sub-threshold level, that may play a role in increasing synaptic recovery. Yeah, we do have plans with that. We're going to be doing some HPLC. What's up? I mean, we haven't really identified yet what is that threshold. We know there is one, so that's part of my experiments as well, is getting some wild type mice and kind of characterizing at 3, 7, 14, 21, and 28 days post injury, what is the global levels of D-serine in the hippocampus. And then from there we can check. Mhm. Yeah, correct. If there's any models that attenuate. Mhm, mhm, mhm. I know we've done like a, that's a good question. I know we've done kind of a inflammatory panel that we found, and we actually found um, there's in our model there's very limited like excuse me in the panel there was inflammatory markers. That's a good question. How do we differentiate that if it's how inflammation is playing a role in that? Just males. No, yeah, just males. So majority. So we looked at for the mechanism for synaptic damage. When we looked at females and what Dena did, when we looked at females, there's actually we did not see the synaptic damage or any of like this PS flipping or anything in females. This mechanism was pretty exclusive to males. So that's interesting. That's interesting. Really, estrogen could play some kind of protective role as a major one. That's yeah. And thank you all for your time.